Welcome everyone to this ARCS extravaganza. We're really excited to be here, all the speakers and myself. My name is Claire Burbick, and I'm going to be the first speaker, which hopefully I can successfully share my screen again after it worked the first time. Okay. Can everyone see my screen? Oh, it's thinking hard. Is that the right view that you're seeing? It is. Okay. Yes. I'm so excited. Things are working for me today. Like I said, my name is Claire Burbick, and my topic today is going to be discussing a little bit about what I do, which is a kind of an obscure field. But my role is in a veterinary diagnostic laboratory. And so I'm just going to talk about kind of the journey that I go on pretty much daily, from what we work at the individual level to this kind of broader focus on public health. I wanted to highlight a little bit of my journey to where I am today, just because many people don't know about this field. And so just to kind of give a little context, I began my educational journey at Washington State University, which is where I'm at currently. So I've been here for a million years and got my Bachelor of Science. I did a number of years of work at the Fred Hutch Cancer Research Center, but my research laboratory was working in HIV immunology. So they do a lot of infectious disease work there as well. And it was a really rewarding and exceptional experience learning about infectious diseases and public health in the human health context. But my true passion is for veterinary medicine. And so I came back to WSU to do my Doctor of Veterinary Medicine in 2004. And then I went into practice because I was like, no more school for me. I have done enough. But then I realized, no, I really do want to still kind of focus on infectious diseases and public health after some practice experience. And so I came back to WSU to do a clinical microbiology residency, which is combined with a PhD. And that's where I became involved with ARCS because I was supported as an ARCS scholar through my residency and PhD training. I was able to get boarded in the American College of Veterinary Microbiology after I finished my degrees, and then did a stint at North Dakota State University's diagnostic lab for North Dakota State, and then ended up back here at WSU, where I'm now an associate professor in the Department of Veterinary Microbiology and Pathology. I'm a section head for bacteriology mycology in the Washington Animal Disease Diagnostic Laboratory. And I also supervise the clinical microbiology residency program, which is what I had completed back in 2014. So kind of a long stretch of academics, and then I'm also back in an academic institution again. And then I like to put in there, like, I am not going back to school anymore. I'm done with that. But what, through all of this educational experience, and then kind of my description of what, my position is, what do I actually do? And I like to say, in academic medicine, we wear a lot of hats. And so I just wanted to kind of take people through what I do and how that works. So, before I go further, full disclosure for people who are squeamish about things, the last slide of my talk does have some more graphic images on them of surgical sites. So just FYI if you are a little squeamish. So to start off, what do I do? So a lot of what I'm involved in is diagnosis of disease at the veterinary diagnostic lab for Washington State. So I wanted to start off with kind of a case to highlight some of the points of of what I do. So this is Dolly Parton. Dolly is a 9-year-old female spayed German Shepherd. Unfortunately, Dolly has had some knee issues, which is quite common in some of our larger breed dogs, and has had right knee surgery as well as left knee surgery. And here's an image of how that surgery works. It's called a TPLO, or tibial plateau leveling osteotomy, where they actually go through with a bone saw and saw through the tibia, realign the angle of the joint, and then put in a plate to secure that bone. So it's kind of an interesting surgery, needless to say, but very common and very successful. Unfortunately, for Dolly, she developed a draining tract, so an infection over the site of that surgery on the left knee. So quite a long time after that original surgery, but is something that we see. She had been treated with antibiotics for a while, had not resolved, and so was referred to WSU to remove that bone plate and to do some further diagnostics, which was to culture the bone plate and determine if there was a bacteria that was causing the problem and what bacteria that was. Unfortunately for Dolly, when we did culture that bone plate, she had a resistant, so a methicillin and a multi-drug resistant Staphylococcus pseudointermedius infection. And so we refer to this type of bacteria as doggy MRSA. Methicillin resistant Staph aureus causes a lot of surgical site infections in people. And so this is kind of our veterinary small animal version of that. So as you can see from our tests that we did, very resistant to a number of different antibiotics. So I just wanted to pause kind of in our discussion to just kind of highlight antibiotic resistance, because it's a big part of what I do. And so really what it is bacteria are exquisitely designed to acquire new genes or to mutate, to be able to allow them to survive all of these toxic challenges, one of which is antibiotic therapy. So they can actually escape the activity and effectiveness of the drugs we use to treat those infections. So you can see from this picture, this is a susceptibility test called disc diffusion. All of those circles are where the drug has killed all the bacteria, which is the lighter part of the plate. And if you compare it to the other hand that this person is holding, you can see that the bacteria have grown all over the plate. And so now those drugs are really not being able to inhibit that growth and kill the bacteria anymore. So that's what we're doing in the laboratory. And unfortunately, the use and misuse of these drugs drives resistance. So we have to be careful with how we use them. Unfortunately, every year resistant infections kill kind of an extraordinary number of people, over 1 million people, but also contribute to the deaths of about 5 million more. And this is in human medicine. We don't have that kind of data in veterinary medicine, but we see very similar resistant infections in our animal species. But we just don't quite have a good understanding of the extent of that at this point. But the big take-home message, and which is a huge bummer for everybody, is that these resistant bacteria are able to spread across many different animal species and humans. And we can also see bacteria sharing resistance genes, which can kind of accelerate that process. So it's a huge issue for us in our ability to practice modern medicine. So not only do I diagnose disease kind of at that individual animal level, so we know that Dolly has a staph infection and it's resistant, but now we have to say, how do we deal with Dolly's infection in a way that preserves our ability to maintain the use of these drugs? And we call that antibiotic stewardship. And so this is our effort to make sure that we have those drugs. So we need to learn how to use them. We need to learn how to not use them, as the quote says. And so a lot of times I'm actually consulting with veterinarians on these cases because they can be very difficult about how to treat the infection and manage the disease. So a lot of times I'm talking about source control, which in this case would be the plate needs to be removed. It's basically harboring the bacteria, and we can't use drugs effectively with that plate in there. I also talk to them about what drugs would be appropriate. So We use potentially different things for soft tissue and bone. Different durations of therapy are appropriate, depending on how extensive that infection is. And then also talking about wound management, things like that. In Dolly's case, she's going to have a surgical site, from the plate removal. We need to be able to manage that so that she doesn't have any problems after plate is removed. And a lot of times it's like, just keep the cone on the dog. Don't let the dog lick the incision, which in human medicine is not really a consideration, but we definitely deal with it in vet med. And so at the end of the day, we want Dolly to be able to go out and destroy things in the yard again. So the other part of that, on top of managing those infections and challenging situations, is we need to actually prevent infection. Dolly has come into our hospital with a resistant infection, and that could have implications for the hospital and for other patients as well. And so my role is to help develop and support infection prevention measures. So Dolly could not go in common waiting areas, could not walk through the hospital. The people, medical staff that were working with Dolly needed to wear, gowns and gloves, etc. She was housed in a special kennel, separated from animals to make sure that there was no contact there. She was basically had disinfection following her wherever she went, and that we had reinforced just washing your hands, which is one of the basic foundations of infection prevention. So, as I mentioned previously, antibiotic resistance is what we consider a One Health threat. So across animals and humans and the environment, because we also use antibiotics in plant agriculture. And so what then starts getting into that bigger picture thing is that we use data that we generate, from our individual diagnostic cases to be able to understand the impact of antibiotic resistance at a bigger level, at a bigger population level. And so this happens across animal and human and the environment as well. And so in our situation in veterinary medicine, we have federal surveillance programs where the state diagnostic labs, which is the map that you see there with the blue dots being where all of the state labs that contribute data to these programs are, allow us to pool all of these individual case data to be able to understand a better the issues that we have in antimicrobial resistance in our veterinary species. And so Dolly's information about what bacteria we isolated and the resistance pattern that we did from that testing is pooled into this database. So as I mentioned before as well, on the veterinary side, we don't have a good understanding of the impact and the extent of antibiotic resistance. And so unfortunately for veterinary things, we tend to have less funding sources available, as well as fewer researchers in this area in comparison to human medicine. But again, to reiterate, those federal surveillance programs have actually brought attention and money and funding to the projects, not only for data generation, but also to actually use the data in these research contexts. And so I've just put up a few of the publications that have come out from us pooling our data and working together as a collaborative across these federal state partnerships to be able to really get our arms around some of this this problem that we have. And lastly, none of this is really, not to say that. It is incredibly important, all of the things that I just mentioned, individual patient care, dealing with infection control in the hospital, the surveillance, and then how we understand the data that we're generating. But we also need to be really, really cognizant of educating around all of these issues. And so that's why I was very excited to come here, is because these are complex issues. They're wide ranging over, all of these different sectors of medicine. And so it's really important for us to get out and be able to say to all of, the people potentially impacted by antibiotic resistance, to have them really at the table understanding and asking questions of people who are really passionate about this work. And so I do education, and I would say at every level that I get an opportunity to, because this is such a One Health community. So this includes veterinary students. I teach bacteriology to the vet students in their second year. I have residents, Clin Micro residents, as well as Clin Path residents, anatomic path residents, internal, all the residents that go along with the teaching hospital, to be able to help educate them as well. We do continuing education for practicing veterinarians. We also help educate the people who are using our lab, with especially if they're ag animal producers or people who don't necessarily have a veterinarian that they're able to access very effectively. And then I do work a lot with my human health partners, because while we have, kind of these different patient populations, it's critical that we're all working together and not against each other, in trying to address some of these antibiotic resistance issues. So I know it felt very rapid. I could talk about this all day, literally, but I want to make sure that everyone has enough time to talk. But antibiotic resistance, and bacterial infectious diseases are just such an incredible health burden across human and veterinary medicine. And our veterinary diagnostic labs, while we're not super well known, are really, really integral parts of the individual patient care that we give to our veterinary species, our dogs, cats, et cetera. But we also are a really important partner in contributing to One Health. And this isn't only just, in this area. We do a lot, we're having a high, high path AI, avian influenza, outbreak that's been going on for a long time. And so we're working across a lot of human animal health landscapes. So the issue that we tend to run into is that we don't have a lot of people who work in this area of veterinary medicine. So the support that ARCS gives for helping to support training of veterinary microbiologists has been really, really critical. And so I just wanted to end with a thanks to ARCS and to my sponsors, Rick and Jackie Doan, who were just amazing support of me well beyond, me finishing my program. So anyways, thanks for listening, and we'll have more time to talk after everyone has been able to speak. We'll stop sharing.
So I believe I'm next. Let me share my screen. Hi, everyone. Okay, I'm having difficulties. So maybe I will just continue without my screen. I had some really cool pictures for you all, but so hi everyone again. Thank you for joining us. So I am a PhD student at UC Merced, and I study soil health and agricultural systems. I particularly look at the management practices, cover cropping and tillage. And hopefully you all know that soils are important to our society, and some of the roles that they play are producing food for us, of course, and some ecological services like water storage, so infiltrating water into our aquifers. They provide habitat for a lot of the microbiology, for the fauna, and for the plants. And then I think one of the more not famous but hot topics right now with soil is soil carbon sequestration. And this is in the form of increasing organic matter input into the soils in hopes that it will be retained there for a long time. So we can do this in the form of compost, manure, biochar is another really important one that we're learning lots about. The one that I'm particularly focused on is cover crops. And this is cool because what we are essentially doing is we are taking atmospheric carbon and we're putting it into the soil plants, into their biomass. And then when these plants are no longer, like when they need to be terminated, they just get reincorporated into that same system, hence increasing organic matter, hopefully long term. So my research is split up into 3 chapters. My first chapter is on soils and evaluating the change in these different management types. This experimental site is really important because it's a long-term, meaning it's been over 20 years of active management. And it's particularly important because it's in the Central Valley in California, which has a very large agricultural economy. And these soils are very representative of the soils along the whole Central Valley. So whatever information we can get from these soils can technically be applied to a large, vast area in the Central Valley. So what we did is we looked at these 2 very stark contrasting management systems. So standard management, which is the implementation of tillage, which is intensely breaking up the soil surface in order to create these nice little beds for the soil seeds. And then we usually leave the soil barren after like our cash crop has been harvested. So yeah, that's standard. And then what we're hoping we will see that is beneficial and that more farmers are likely to implement is conservation management, which includes minimal or no disturbance of the soil surface, and the addition or the application of cover crop, which is just a non-cash crop that is added sometimes in the winter, and it's usually started up with just rainwater, but if it's not adequate, then we use like irrigation once or twice. So it's, hopefully very low water use. So what we looked at was basically we were just adding to a trajectory of these 20 years of what's going on in the system. So we looked at carbon and nitrogen, and we found that these systems double the amount of carbon and nitrogen. So that could lead to maybe like less inputs of synthetic nutrients. And then we also looked at some physical properties of these soils, and we found that in the conservation management, we had a lower bulk density, which essentially means that these soils are less compacted, which is good for like airflow and water flow, and especially good for plant roots. We also looked at the stability of the different clods of these soils, and we found that in conservation management, these clods of soils were much more stronger. So it leads to like chunkier soil versus like very granular soil that it doesn't really hold water as well, and other things of that sort. And then lastly, we looked at the carbon of the soil. So not just the quantity, but we looked at where the carbon is located and the chemical characteristics of this carbon. And this is important because location and carbon characterization can determine how long a soil or how long a carbon can be in the soil. So we determined that in conservation management, we had more carbon that was minerally associated or like clings on to these minerals that essentially kind of make it really difficult for microbes to access, to break down, and then respire as CO2. And then in terms of characterization of their carbon type, we found that the conservation management had slightly higher complex carbon, which also means, again, that it's more difficult for these microbes to access. So essentially, we see this continued improvement in increase in chemical properties like carbon and nitrogen, as well as these soil properties that include like better air diffusivity, better water movement, but then again, at the same time, better water retention in some of these smaller pores. So we were curious, like, how will this impact carbon mineralization? So in my second, I don't know where my slides are at this point, but in my second chapter, what we did is we took some of these soils and we saturated them. And as they went from saturated or wet to dry, we took multiple respiration points or carbon flux, and we determined something very interesting. So in conservation management, we inadvertently create this environment that causes accelerated carbon loss. And the scary thing is that a lot of soils and agricultural systems are kept at this like very specific moisture range where we have accelerated carbon loss. So this really points out the importance of looking at soil as like a holistic system where we're trying to do kind of like a balancing act of increasing productivity, increase in carbon storage, and then maybe in a way being able to decrease the amount of water that we use. And then, so for my last chapter, I'm happy about my chapter, but I'm kind of sad because the third chapter arose from the unfortunate situation where this research site no longer could manage the fields anymore. So they stopped or they fallowed the lands, which is a common agricultural practice that a lot of agriculturalists have to do when there's no water or when soil becomes kind of like barren or just not workable anymore. So we're gonna do the same exact analysis, like look at carbon, look at nitrogen, look at the stability of the soil, to in a sense look at the potential or the resilience of these soils to keep these elevated properties, or just to kind of give an idea to the local farmers, like what happens to their lands in these different management sites when they apply fallowing. Fallowing could be something that we see more often in the future. So we're really excited to provide that information to farmers in the area. And I just want to thank ARCS for their support. A lot of the work that I did for all of these chapters actually was done with the help and support of ARCS, getting me to move or helping me move to Merced to do all this intensive lab work. But yeah, thank you.
Thanks, Jennifer. It's my turn to go over my research. Hopefully my screen shares properly. Can I get a thumbs up from the other panelists if you can see this? Oh, fantastic. Hi, my name is Arabi. I am a 6th-year PhD student at UC Merced, and I'm also concurrently a Fulbright scholar, and I'm based in Paris right now, so I'm calling you from just west of Paris. And I put a big Eiffel Tower on my slide because my current daily commute, I take a bus past the Eiffel Tower every single day. So it's genuinely such a dream to be out here. And I really want to first take a moment to thank ARCS, because I would not have been able to move to Paris if it wasn't for the scholarship. The Fulbright is supposed to be this joint scholarship between the French government and the American government, but for some reason, when they awarded me the Fulbright scholarship, it was less than the minimum wage in France. And so I was having a lot of trouble getting a visa. And then I was awarded the ARCS scholarship afterwards. And because I had that supplementary ARCS funding, I was actually able to move here and conduct this scholarship. And also just, you know, with Doge cuts, actually all of the Fulbright staff got furloughed, and there was a little bit of concern if we were going to get stipends. It was paused for a few months. Everyone, I believe, now has had their stipends, but luckily I didn't really have financial concerns because I also had the ARCS funding available to me. So just a huge thank you to start off with. To go over what my research is, I am a computational condensed matter physicist. And what I study, sort of a central theme in my study, is looking at new materials that can be used to create quantum bits. And quantum bits are part of this like thing, quantum computing, this hot button topic everyone is hearing about. And I just wanted to give a little bit of an overview of what that kind of means. So on the left here, this is a traditional bit, like a schema. So if we look at regular computers, just even your phone, everything is encoded into a bit, and a bit has sort of two options. It can either hold a 1 or a 0. So if you have, for example, 10 physical bits, it would hold 2 to the n amount of information. So 10 physical bits would hold 1,024 pieces of individual information. Well, a quantum bit says, what if we can create a bit that holds more pieces per bit? So one of the schematics I'm looking at is, what if I had a 3-level bit? So it had a 1, 0, and a -1 option. So now each physical bit would hold that much more information. So for example, a 10-bit system that has 3 options would now hold 59,000 pieces of information as opposed to 1,000 pieces of information. So in the same physical space, you could hold that much more computing power. So it would just supercharge computing if we could get this to work. Just anything that would use a computer would be that much more accelerated. So any field you can think of, supercharging AI. I know people aren't really excited about that, but we could do that. Also, medical diagnostics, anything that required a lot of computational power would just have that much more support. So this is what I'm trying to find, a material that would be useful as a quantum bit. So these are a couple of the things that I'm working on. a big overarching topic in my research is looking at 2D materials. So if you think, I had a friend say this to me earlier, a water bottle is like 3D, but then a piece of paper is 2D. It's just, technically paper is 3D too, right? But it's just completely flat, like essentially just one layer. So that's what I'm looking at. 2D materials are materials that are essentially just a layer thick. And on the left, I'm showing research that I have at UC Merced. So we have these experimental collaborators where they can create the single layer of WS2. And then in lab, just in the process of creating that material, it has little holes in it just naturally whenever you move something. So what they figure out how to do is they deposit cobalt atoms on it, and they use this STM tip, that's a type of microscope, and they play golf with the cobalt atoms. So like the atoms just naturally get nudged into these little holes, and we create a new material. This is called cobalt defected WS2, and they've asked us to computationally model the electronics of the system to see if we can actually turn this into a qubit. On the right, I have sort of a similar thing, but this is what I'm working on here in Paris. So at the University of Paris-Saclay, I'm looking at 2D materials with the magnetic field pointed through it to see if that changes its properties, its electronic properties, to make it a possible qubit. And so what I'm showing here is a plot that I made where when you have the magnetic field in it, how do the electrons redistribute in space? And so that is sort of what I'm working on right now in my projects across the world. Ultimately, it's going to go towards applications in quantum computing, but honestly, I just really feel like I'm uncovering secrets of the universe and like learning fundamental physics. So I want to thank you again, everyone who's involved in ARCS. This scholarship has helped me in so many ways. And with that, I'd like to pass it off to Sarah.
Arabi, can you unshare your screen?
Sorry, I'm trying. Technical difficulties.
You keep telling me I can't do it up there. All right, thank you. Okay, now let's try this. Okay. Is this visible? Fantastic. Okay. Hi, everybody. My name is Samuel Carey. I'm a fifth-year PhD candidate. Quickly threw this slide deck together, so it's not perfect. I apologize. I am defending in a month, actually, so my priorities are dissertation stuff. But quick background about myself. I am, a human movement scientist at heart. I started at Michigan State University studying kinesiology, and I worked in a lab with a wonderful advisor where we studied stroke rehabilitation, movement dynamics, and integrating technology and behavior. Transitioned into my master's at the University of Massachusetts Amherst, where I got more into full body dynamic movements of how are people able to walk and reach and grab stuff at the same time, and how is our brain able to integrate all of these types of behaviors. And then finally got to the University of California, Merced, where I got to pursue everything I've ever wanted with another wonderful advisor. I now work at the Sensory Motor Neuroscience Lab in the Cognitive and Information Sciences Department, where I study basically that didn't like the transition. Terrible slide. Human sensory, cognitive, and motor integration, and I primarily look through postural stability. Beautiful. So what I essentially look at is how are humans able to exist in terms of their human behavior? Human behavior is extremely complex. It seems simple on the surface. We don't have to think about a lot of the stuff that we do day to day as picking up a pen and writing your name or grabbing a cup. However, there's a bunch of different pieces that go into play in order to create human behavior and allow this behavior to transition between grabbing a cup, taking a drink, all while not falling over. And that's kind of what I use as my medium, is I look at postural control. Now, most people, when they think of postural control, they think of just how straight your back is. A lot of people nowadays, they think of, having good posture. It's keeping a back straight, keeping your spine in alignment. And while that is correct, Postural control is so much more than just having a straight back. It is your body and your brain's capability and performance of keeping you upright and stable. If we didn't have postural control, you would be falling over constantly. And to think about postural control, think about how people just stand upright. Seems like a simple task, but on the motor and the neurological level, if you try to balance two rulers on top of your fingers and then put a ball on top and keep all of those things balanced in perfect synchrony, that is a simpler task than your body's standing up just as you would at your desk. And there's a bunch of different ways our body is able to perform this supposedly simple task. And a lot of that comes down to the musculoskeletal system. If you have a lot of muscles constraining these joints, these areas, your brain is extremely good at what it does and is able to send the proper signals to activate, coactivate, or inhibit muscles at not light speed, very fast neural responses. And we're great at it. Humans are fundamentally good at staying upright and staying stable, or so we think. In reality, our environment around us is extremely complex, as is the human behavior and the human body, and everything in our environment also basically interacts and interferes with our body's ability to stay standing, to stay stable. Everything from simple sensory environments of a car driving by if you're walking on the road, or you performing mental math in the grocery store as you're working through how much your bill is going to be. All of this will impact your body and your brain's capabilities of just staying upright. Otherwise, you'd be falling over, you'd be tripping, you'd be falling. And that's basically where all of my PhD research kind of comes into play. I look at how our sensory environment, our cognitive neural processes, and our human musculoskeletal system all communicate and work together to maintain what we perceive as the simplicity of existing in human life, of just being able to sit at your desk or stand up and walk to get a cup of coffee or talk to a friend, whatever it is. All of these behaviors can exist because your body's able to remain stable. So a lot of my work exists in a lab where we use what's called force plates and motion capture, which I'll show on the next slide. But we basically look at how your body just moves, these very small movements in space, and it kind of creates this center of pressure mapping. And if anybody has seen Hollywood or anything where there's aliens, zombies, monsters, Lord of the Rings is a great example of Smeagol. Motion capture is a technology that is used across multiple different domains. Hollywood is one of the more famous regions. You see on the picture to the right, you see these kind of like glowing balls. All these are just small little spheres that we can place over the course of the human body. And then with our 8 camera, I think we now have 10 cameras set up in the lab. We can then create a 3-dimensional live feed of your musculoskeletal system in space and time. And we can track basically everything that your body does in response to the environment around you. And the first half of my PhD, I decided to look at the sensory environment, how sensory stimulation can impact stability. And all I did is I gave my participants noise, just static through an auditory system. So gave them a pair of headphones, had them listen to white noise. And we found out that just by listening to white noise, humans become a lot more stable and, more importantly, adaptable to their environment. So your stability basically just increases through the simple act of listening to white noise. And there's a couple theories that we're still playing with as to why this may be. A big one is what's called stochastic resonance, which I don't think I have enough time. Again, I can talk about this for hours on end. But it was great. We found that just listening to white noise can help you become more stable. And then we wanted to push it further. Auditory is great, but what about the tactile modality? So we had them wear this backpack. It was kind of like, those like hiking camelbacks, just very smooth. It fit along your spinal column and it vibrated and you couldn't feel the vibrations. But same thing, we introduced this white noise, slight vibration to the spinal column and we elicited the same effect. Just by your spinal column receiving slight vibrations that you didn't even really know were happening, your body became more stable and adaptable to your environment. So it was awesome. We found that stability is impacted by the environment, which we knew, but we were able to physically manipulate your environment to help you become more stable. Then I wanted to push it and look at the cognitive system. So how does thinking about something change your postural behavior? And we did this within my lab. We have our bread and butter is what's called a transcranial magnetic stimulation device. And what this is, short for TMS, is this copper coil wand that we can create an electromagnetic force and send a pulse into your cortex, so the surface region of your brain. And I'm paraphrasing, but we can turn off regions of your brain. We can simulate synthetic lesions for short durations, only lasts about 45 minutes before your brain returns to normal standing. But with this machine, we're able to alter the electrical activity of specific regions of your brain to see how this may influence other behaviors. And for me, looking at cognition, I decided to focus on the frontal cortex, which is primarily where working memory is kind of thought to exist. So what I did is I had my participants come in and I just turned off their working memory system, which theoretically should not impact postural control because that's all cerebellar. However, just by altering their working memory system, having them come to the lab and stand, they performed various tasks, but across the board, just by inhibiting your frontal cortex, we found that your postural control system was diminished. You became less stable. Your body moved in a more chaotic pattern. And we had them perform various cognitive tasks on top of this. And we validated that by turning off this region of your brain, we worsened your ability to perform the cognitive tasks, and more substantially, a lot of participants, we talked to them afterwards and felt this kind of risk of they thought they were going to fall over just because they were standing there and their working memory system wasn't working appropriately. So with all of this work, I've been doing countless projects over the last 5 years. What we're starting to discover is that there is this constant interplay between the human sensory, cognitive, and motor systems within the human brain and body. And historically, within my field of study, people like to separate these regions. They look at just the motor system or just the sensory system, just the cognitive system, and ignore everything else. And while that is great for certain types of research, what I'm trying to kind of push with my idea is that you can't actually do that because all of these systems are constantly communicating and interfering or accelerating each other's behaviors to create what we know of as human behavior. And currently I'm writing it all up in a dissertation. So if you want to find out the whole story, you can read that. It's great. But I just wanted to thank ARCS for all of the help this past year, being a fifth-year PhD student, trying to finish everything, write a dissertation, defend all of that. It's chaotic, as I'm sure Jennifer and Arabi are very aware of, and everybody else that's been to grad school. And the ARCS Foundation has been a massive help in just keeping me afloat and keeping me pushing forward, helping with financial burdens that all grad students face, and just making my life so much easier so I can actually focus all of my attention on my research. But yeah, thank you very much. And Claire, back to you.
Thank you. So thanks everyone. It's fascinating work. Yeah, I have so many thoughts now about all the things. But so I guess for those in the that are listening, if there are any questions that you have, we'd love to hear them. You can put them into the Q&A area, or the chat probably will work as well. I think we're monitoring both. But I guess I can start off with some, I guess, a question. So I think, as we go through, graduate school, it is obviously a huge challenge, and, the areas of study are obviously incredibly challenging and exciting as well. But I guess, kind of one of the things that I guess I would be curious about, and was kind of suggested as a jumping off question, was like, what are the fun parts? Because I think there's a lot of, grind and whatnot to trying to get through these programs, but I think, that's always kind of the counterbalance to it is, what kind of drives you and what excites you and what's truly enjoyable about about what you're doing.
I'll
So
go.
if anyone wants to go, yeah, Arabi, go for it. Yeah.
Robbie.
I'll be short. Specifically in Merced, I think it's a really, really small grad program. It's about 700 students from what I understand. And just, it's really, really nice to have a small community where you know everyone and you run into your professors at the grocery store. And it's a really supportive environment, I think, for everyone. And I do think that gets lost a bit in bigger schools, but we're really lucky to have that, I think.
So yeah, to further that, it like in my department, it's great because you just like, get up and walk around, just the floor, and you run into other grad students and get to events, push your ideas that you're currently thinking about, but like having that community around you of like people who are all in the same bank, but you're all there for the same reason, and that is like your passion for research and like pursuing those ideas. And it's for me, it's always great. Like there's the grind, there's the difficulties, but having that aha moment, like when your code finally works and you get your analysis and like, that big moment at the end is just, for me, makes it all worth it. Like I absolutely live for those moments.
Yeah, I'd have to agree with both Sam and Sarabi. The community is quite small at Merced. I think our campus is, I mean, it's been growing for sure, but even our campus is kind of small. And on our floor alone, we have 3 soil labs, or like ecology, agroecology labs. And it's just really exciting to be able to like kind of walk just down the hall, and there's like a whole other lab with like equipment that the other PIs are like more than happy to share. And then you get to like kind of just peek in on your other colleagues like work, and then it kind of just inspires you like, hey, like I didn't think about doing that, like maybe I should do that. Or like yeah, if you need help with code, or like, hey, I know you did this analysis before, like do you mind like sharing your code or like walking me through it or something. And it's been really great, very collaborative work, very, very.
Yeah, I think, obviously I finished my graduate training a while ago now, but I think it held true for me too. Like all of what you're describing is just sort of being in that environment of learning and the diversity, I think, of what people are doing is so inspiring. And I think in all honesty, that's a lot of why I stayed in academia, is because I really did enjoy being around the smartest people I've ever met all the time, and just being kind of overwhelmed with what people are sort of intellectually capable of across like a really kind of wide thing. And then also finding like, hey, I think this person could really, like, even though they're in a very different field, like would really collaborate well with what I'm doing too. So I think there's just really unique opportunities that kind of drive. I don't know if that would be considered fun, but I think it's a pretty amazing part of that whole experience, especially being in an academic institution. And I think it's just, it really is almost kind of just this like not a self-fulfilling prophecy. I don't know, whatever that is, where we're building on top of each other and like making each other better, just kind of being in that environment. Oh, it looks like we have some questions. Okay, so we'll go with does the soil research have applications for home gardeners, such as using cover crops in vegetable beds?
I guess that's for me. So in terms of using cover crops, I don't think in home gardens, I feel like in home gardens, I mean, I would imagine that people are constantly changing their crops based on like seasonality, versus it maybe in more of like the like large scale like ag systems, like they maybe just specialize in like 1 or 2 crops. So that's why I feel like cover crops are probably not as useful. But then again, yeah, I guess so some cover crops are actually known for like nitrogen fixing abilities. So I guess, yeah, in a sense, if you feel like you don't want to use money on like expensive organic matter like amendments, or even like synthetic fertilizers. I guess cover cropping could probably essentially help with some of the nutrient aspects of your small at-home gardens. And then some cover crops are really good too at remediating like compaction, and they have like really deep roots and things like that. So actually, yeah, it could help. Yeah, I could definitely see it help. Yeah. I don't know if that answers your question.
So I think this is kind of a question for everyone. So what inspires you most about your work? And what do you most look forward to in impacting in your field?
Big question. I think for me, like the inspiration came from my undergrad when I first started in research, working with stroke rehabilitation, like being able to see the application of my work directly to somebody who needs it, like really kind of got me going in my research. And I've always kind of like had, some stake in the clinical applications of my research, which keeps me kind of going day to day. I don't really do clinical research anymore at UC Merced just because smaller school, there's not really the hospital attached to it. However, it is coming. So in the future, there will be clinical research. And I'm so excited for that, for the future grad student generations to get on that horse. Moving forward with mine, I just like I want to see my work actually get put to use. Like I'm starting to discover capabilities of increasing stability so that can be given to older adults who have high risk of falls, people with neurological disorders who have instability, stuff like that. Something as simple as white noise can help make their day a little bit better, stuff like that. And I have a current first year grad student who's taking over kind of like the horns of my research and now starting to look deeper at the neurological side of things with electroencephalography, which is this head cap where we can actually look at what's happening in the brain when all of this stuff occurs. So I'm really excited for them to continue their research and look deeper at kind of my original findings and see, what's actually going on under the hood.
For me, it's a little bit different. I'm really interested in physics and research, just sort of for the sake of physics and research. I'm really, really into this like abstraction. Like I really think of physics as like poetry or something. Like I really just find it beautiful. And that's sort of what drives me, even as we were saying before, honestly, doing a PhD is really, really hard. And even if you love the project, it's 5 years and 6 years in, it gets so rough. But it's sort of just like this higher thing that keeps me going. And then for the future, what I really, really want to do, I'm like, my goal is to become a professor and have a research lab. And similar to what Sam was saying, like I really want to have students and show them how to do research and watch them grow as researchers and sort of have everyone participate in this thing that I think is really beautiful. So yeah.
So I don't have any published work at the moment. It's really hard to get published. And I think the only talk that I'll be having soon is hopefully my defense. So I don't know if ARCS shares information like that, or we're always, I think after COVID, a lot of us just automatically send out Zoom links to our defense. So I'd be happy to have any of you go to my defense and learn more, get the final results for that one following chapter. And then I did see that there was another question in the chat regarding the soil research that we do at my lab. My lab research is mostly focused on soil physics, so the movement of gases and water throughout soil. And before I think I arrived to my advisor's lab, he was very just like theoretical work and didn't really apply it too much to like, application wise. So I don't know, maybe that could be a potential research for him in the future, but as of now, it's mostly just theoretical work and agricultural systems. Yeah.
Sam, you have a kind of a question targeted at you. Have you seen any positive results using EMS?
Ooh, so TMS, hopefully that's just a typo.
Q.
No, you're good. I just looked at it. So TMS is just a mechanism that we can use to alter the brain's electrical dynamics. You can flip the polarity of that coil, and in my research, we were down-regulating, so we were turning off certain regions of the brain. However, TMS right now is being used in clinical research for mental health, and it's actually being shown to, we can get deep brain stimulation with TMS at a certain frequency and polarity where it helps increase what we call the plasticity of the brain. So a lot of time with people with, I'm not a mental health expert, it's just reading the literature and just knowing people that work in it. But with people, for example, like depression, your neurons get stuck kind of like in a well. They can't produce the hormones that you want to produce. And with TMS, we can basically go in and alter the neural activity of those regions that are supposed to produce serotonin, dopamine, whatever it is. And TMS actually right now is, we're finding great results on the clinical applications of TMS for positive benefits for people with mental health, PTSD, schizophrenia, stuff like that. So it is on the up and up. It's becoming great, but it is a very new field of research. So it's going to be a couple years before we see any kind of like actual output outside of a research lab, that kind of stuff. But TMS can be used in a positive way, not just turning off parts of the brain like I do.
Did anyone else want to comment on any presentations, other resources, etc., that are on the horizon that you could share?
Like Jennifer said, getting published is really hard. So I have I think I have like a second and a third author paper out that can be like Googled, but I don't think that it would be very elucidating, and it's definitely not my primary work. And like I would, if anyone wants to ask me questions, honestly, like feel free to send me a message. I'd be happy to talk about it. I think actually even asking that question is such a good point. I think something that we should all be doing is like making websites with like our research forward so people knew what we were doing. But I think right now it's kind of all obscured.
Yeah, I can add, my advisor does a really good job at posting up our publications on their website. So I could definitely add that into the chat if anyone is curious about the work that we do at a soil physics lab.
Yeah, I think for myself, most of my, I have various presentations that I do kind of for our, various professional organizations. And then I do have a number of publications. I think in my field, it's a lot easier to publish than in certain other fields, which is kind of a bummer. But yeah, I'm happy to share, publications across sort of the vet diagnostic lab spectrum and looking at some antibiotic resistant stuff as well, if anyone would like is interested in that, which are I think a little bit maybe easier to digest than some of the other speakers' fields, which are very, very complex. Not that antibiotic resistance isn't complex, but I think yeah, I think it maybe is a little bit more in lay terms than some of the other topics too, but happy to share as well. Oh, so we're right at 1 o'clock. And I think that was all the time that we have for today. So really appreciate everyone being here and such interesting areas of work for the speakers. It was just fascinating. And so yeah, with that, I think we'll close it. And thanks to everybody.